Homeostasis. Homeostasis is essential for the proper functioning of living organisms as it maintains the dynamic equilibrium of internal physicochemical conditions. This delicate balance ensures that crucial factors such as temperature and pH are kept at optimal levels for metabolic reactions. Enzymes, which are primarily composed of proteins, play a vital role in these metabolic processes. The structural configuration of proteins is crucial for their enzymatic function, and factors like temperature, pH, and other environmental conditions can influence it. Consequently, the ability of living organisms to maintain a physico-chemical balance throughout their bodies and within cells is imperative. All organisms, from unicellular entities to complex multicellular organisms, actively maintain homeostasis. Within cells, specialized pumps help expel waste products and regulate water content, contributing to overall internal stability. While plants, fungi, and simpler organisms may not be able to regulate their body temperature, they have compensatory mechanisms that allow them to thrive in diverse environments. In contrast, animals can be categorized based on their capacity to regulate body temperature. Fish, amphibians, and reptiles cannot regulate their internal temperature and rely on the external environment for thermoregulation. On the other hand, birds and mammals can regulate their body temperature and exhibit sophisticated adaptive behaviors and anatomical structures. For example, birds and mammals have structures like large ears that efficiently dissipate heat, helping to reduce body temperature. Conversely, behaviors such as shivering can contribute to the elevation of body temperature. These examples highlight the intricate strategies organisms employ to navigate the complexities of their environments and maintain homeostasis despite varying physicochemical conditions. Response to stimuli. Response to stimuli refers to a change or reaction in an entity triggered by either an internal or external stimulus. My biology teacher, Dr. Ashok, often illustrated this concept with an example that has stayed with me. In his paraphrased words, consider an elevator. Pressing a button on its panel causes it to move up. It does not matter who presses the button. It could be you or a donkey. The elevator simply responds without discretion. This example underscores a mechanistic reaction to stimuli devoid of judgment or complexity. However, with advancements in artificial intelligence, artificial systems now exhibit responses to stimuli that closely mimic human intellect, showing remarkable adaptability and context-based actions. In living organisms, stimuli can originate from both external and internal sources. External stimuli include changes in temperature, humidity, or rainfall, which elicit corresponding physiological or behavioral responses. For instance, plants bend toward light, phototropism, and animals seek shelter in response to rain. Internal stimuli, on the other hand, arise from within the organism, such as the release of hormones or intense emotional drives like obsession. These internal triggers often guide processes like growth, reproduction, or even complex decision-making in higher organisms. For example, the developmental transition from tadpole to adult frog is regulated by internal stimuli, specifically the release of thyroid hormones. These hormones trigger a cascade of genetic and physiological changes, such as the resorption of the tail, the development of limbs, and the formation of lungs, enabling the organism to adapt to terrestrial life. This entire process is driven internally, independent of external signals. Similarly, male deer naturally shed their antlers annually due to a decline in testosterone levels, an internal stimulus that regulates this developmental process. The reduction in testosterone causes the weakening of the connective tissue at the antler base, leading to their detachment. This is a programmed event that occurs cyclically without reliance on external factors. The responses of living systems to stimuli are multifaceted and context-sensitive, making them unique and dynamic. Motility Tesla cars demonstrate autonomy by transporting individuals from one location to another. However, they still require human intervention to recharge their batteries, and humans program their decision-making capabilities. In contrast, living organisms exhibit inherent autonomous mobility, showcasing a variety of motility mechanisms driven by survival, nutrition, or response to environmental conditions. When faced with threats or seeking sustenance, living organisms display diverse types of motilities. Bacteria and sperm use flagellar motility, utilizing whip-like structures for propulsion. Protozoans like amoebas form transient cellular projections to guide their movement. Organisms like Hydra somersault toward their destination, while earthworms move through bodily peristalsis. Snakes utilize rectilinear locomotion, while fish use fins to propel against water. Amphibians, insects, and mammals, including frogs, primarily use legs for movement. Birds have evolved wings for flight, showcasing remarkable evolution. The octopus, a highly versatile organism, exhibits a wide range of movements based on its requirements. It can walk on the ocean floor, propel itself through water, and even glide, demonstrating an impressive repertoire of locomotive abilities. The common thread among these diverse motility strategies is the response to stimuli, a fundamental feature of living organisms. Unlike the programmed responses in autonomous vehicles, living organisms engage in dynamic decision-making, driven by a combination of prediction, planning, and strategic responses to their ever-changing environments. This adaptability underscores the complexity and sophistication of the inherent autonomy observed in living organisms, starkly contrasting with the programmed nature of artificial systems like autonomous cars. The absence of a cell wall in animals is fundamental to their ability to achieve locomotion. While a cell wall is vital for structural integrity and protection in plants, fungi, and certain other organisms, it imposes rigidity that limits flexibility and coordinated movement. The lack of this rigid barrier in animal cells allows for remarkable mobility characterized by flexibility in multiple dimensions, ranging from the number and direction of movements to the degree of motion enabled by interactions with external substrata, such as surfaces or levels. This capacity for locomotion in animals is the cumulative outcome of several factors. First, the absence of a cell wall permits greater intercellular connections, allowing cells to work in synchrony. Second, motor proteins such as actin and myosin play a pivotal role in changing the shape and size of cells, often producing dramatic effects that translate into coordinated tissue and organ movements. Third, integrating these cellular movements with the nervous system ensures precise, purposeful motion. The nervous system orchestrates muscle contractions, coordinates responses to environmental stimuli, and fine-tunes movements to optimize survival, reproduction, and interaction with surroundings. These adaptations enable animals to exhibit dynamic, impressive movements, running, swimming, flying, or crawling. The intricate interplay between cellular flexibility, molecular mechanisms, and neural coordination underscores how the absence of a cell wall, far from being a limitation, becomes a defining feature that empowers the diversity of animal locomotion. Genetic information. The most common characteristic all propagating entities share is the possession of genetic information. This genetic information is carried by nucleic acids, with DNA being the predominant genetic material in most propagating entities. However, there are exceptions, as certain viruses and viroids use RNA as their genetic material. Cellular entities typically have double-stranded DNA, which forms the genome. In contrast, non-cellular entities can have double-stranded or single-stranded nucleic acids. The genome represents the collective genetic material of a cell and contains the instructions for producing proteins and RNA molecules. These molecules play crucial roles in carrying out structural and enzymatic functions necessary for the cell's objectives. A fascinating example is mammalian red blood cells, which lack a nucleus except for camel's RBCs, but maintain stable mRNAs to replenish the protein pool as needed. Platelets, although debated as cells or not, are essential components with critical bodily functions. They do not possess genetic material, but do have mRNAs. This unique characteristic allows platelets to respond to stimuli, secrete components discreetly, and even take up glucose from the blood. The translation of these mRNAs results in the production of specific proteins crucial for platelet function. The organization of living organisms is characterized by inherent complexity and strategic planning. Basic life properties include possessing genetic information, existence, the ability to change the environment, and reproduction. Higher life properties encompass more intricate features, such as the response to stimuli, active acquisition of energy and matter, multicellularity, instincts, sexual reproduction, spatial and temporal planning, longevity, and a disproportionate amount of genetic information. These properties collectively define the remarkable complexity and adaptability observed in living organisms, showcasing their dynamic interactions with the environment and their strategic approaches to survival and reproduction. Death. Have you witnessed the death of a dear one? A death that is long and almost longed for? Have you experienced their breath growing shorter and the intervals becoming longer? The heartbeat dropped until it stopped while his hand was in mine. My father's death, after a year-long bedridden life due to paralysis and cancer, was the most depressing and perplexing view of life and death. Is death a loss or a relief? That was my peak period of drowning in depression and waking up to philosophy. Death is the point at which life ceases to exist as an autonomous entity. Different organisms experience death differently, but it marks the beginning of the end. All organisms are susceptible to death due to external forces or internal vulnerabilities. External forces, such as natural disasters, accidents, or conflicts with other organisms, can result in death. Infections, diseases, and lack of food, water, or air can also lead to death. Additionally, in some cases, death may be self-inflicted, as observed in human suicides. In most organisms, death eventually occurs even under favorable environmental conditions and with abundant nutrition. This is known as natural death. Aging or senescence, which precedes and causes natural death, refers to the process of an organism growing older with time. Natural death is typically the result of the organism's inability to assimilate nutrients, eliminate toxic byproducts, or generate new cells to sustain the functioning of organs. However, natural death theoretically does not occur in organisms such as bacteria or similar organisms that reproduce through binary fission, where one cell divides into two cells. Natural death theoretically does not occur. Instead, they continuously reproduce, giving rise to new entities. Similarly, some multicellular organisms like hydra, sea anemones, and starfish can reproduce through division, creating new organisms without the concept of natural death. In these cases, the parent entity ceases to exist. It becomes a part of new entities without an intermediate stage where it is not a part of a living organism. This contrasts with complex organisms, where an entity ceases to exist, degrades, and becomes part of the material cycle in the biosphere. Natural death is inevitable if other forms of death do not manifest in organisms that exhibit segregation between somatoplasm, the body responsible for individual survival, and germplasm, the reproductive cells ensuring species continuity. This distinction underlies the mortality of somatic cells, as they exist solely to support the organism's life and reproduction. In contrast, the germ cells perpetuate genetic information to future generations. Aging, a precursor to death, is a progressive decline in physiological function and cellular integrity. One major factor contributing to aging is the shortening of telomeres, the protective caps at the ends of chromosomes. With each cell division, telomeres progressively shorten due to incomplete replication, eventually reaching a critical length that halts further cell division, leading to cellular senescence or apoptosis. Additionally, the accumulation of genetic mutations over time compromises the efficiency of cellular processes. Nuclear or mitochondrial DNA mutations can disrupt protein synthesis, energy production, and other essential functions, hastening cellular and tissue degeneration. The exhaustion of adult stem cells in one or more tissues is also a significant factor. Furthermore, the gradual buildup of metabolic waste products, such as damaged proteins and lipids, overwhelms cellular repair mechanisms. These waste materials can impair cellular function and trigger inflammatory responses, further exacerbating tissue damage. You can't get out of life alive, so you may as well have a good time. Les Brown.